Good afternoon.
Thank you for joining us in this first lecture of a series on mathematical biology. I will leave the floor to Professor Touma, the director of CAMS, to introduce the series. The floor is yours, Professor Touma.
Thank you, Fatima, and thanks to Professor Saad for joining us for this first lecture in the series. I want to first start by thanking Dr. Mroué for leading on this effort, practically single-handedly, to try to reflect our center's interests in mathematical biology, primarily in terms of the beauty of the field in itself, but also in terms of its implications for the context in which we're working. We're part of a university with a stellar medical school, which is also neighbored by other universities with medical schools, each involving high-level research as well as clinical practice, and each benefiting potentially from the contribution of mathematics and mathematical modeling for their work. We're particularly excited in this context in terms of an initiative which we are pursuing with the medical school at AUB to catalyze and strengthen the dialogue between mathematics, mathematical modeling, data analytics, as pursued and featured through the work of CAMS and the work that is being done in the medical school. And in that context, myself, Fatima, our colleagues in mathematics here at the AUB in biomedical engineering, with whom we've had a long-standing collaboration through CAMS, as well as our colleagues in the medical school, are very much looking forward to the lectures as an occasion to basically start having this conversation around the issues that are of current and important interest. So Fatima, thank you again for leading on this effort. It's been a pleasure working with you, and I very much look forward to what you have managed to put together for us.
Thank you. Thank you. It's my pleasure to introduce Professor Mazen Saad, who's a professor of applied mathematics at the engineering school École Centrale de Nantes, where he's the president of the scientific council there since 2017. He's also a member of the Federation of Mathematics, and the National French Council has attributed to Professor Saad the prime for his excellent scientific research since 2013. His research interests focus or are concentrated around multi-phase flow in porous media, modeling in biomathematics, and on finite volume methods. Thank you for being with us, Professor Saad. Please, the floor is yours.
Okay, thank you, Fatima. I would like to thank firstly Dr. Fatima Mroué, the organizer of this talk series. Fatima asks me to give an overview of some of my research on biomathematical modeling, and for a large audience. I have tried to remove the technical mathematical results about existence of solution or something like that. And I focus my presentation on the modeling of some biological process and how to translate them into mathematical equations and how to solve them. Nevertheless, I have to give you in my presentation some equations or operator modeling biological phenomena. Okay, for the outline of my presentation, I'll introduce the process of chemotaxis, and I'll give you some examples to illustrate the process, especially the model of Keller-Segel. The rest of my talk is a succession of examples. Okay, I give you here for for each example, I will specify the biological process and the mathematical model, and some numerical results will be presented. The first example concerns bone healing. The second one is about the breast cancer initiation. And the third one here aims to understand the brain cancer, the glioblastoma. And finally, in the context of personalized medicine, how to calibrate the biological parameters with the help of data as MRI and some analysis of patient. Okay, let us first define what is chemotaxis. Chemotaxis can be defined as directed movement of cells and organisms in response to a chemical signal or a chemical gradient. Chemotaxis is the ability of organisms and cells to immigrate through the detection of chemical signal and to adapt their movement accordingly. This behavior of cells enables them to locate nutrients, avoid predators for animals, and it can be an attraction or repulsion. I show you here only in this my talk one video, so I have to stop share here. Sorry. Okay, to illustrate this mechanism, we can see in this video that is here a pipette swapped by a chemical species, and here this organism or multicellular organism detects the chemical, and then it modifies its displacement according to here the high concentration of the chemical. And we see here also another organism who are attracted by this signal. Chemotaxis is a vital characterization of living organisms to adapt their movement for following the signal. Okay, I switch now to my. And chemotaxis, or we say also haptotaxis. Haptotaxis is where the signal is not in full read, perhaps as enzymes. We say here about haptotaxis. Play an important role in many biological fields, and in especially here what we see in the growth cancer, the bone healing, etc. The first example here is given by the bacteria Bacillus subtilis. This bacteria can be found in the ground. The bacteria here move towards high concentration in oxygen here to survive. The nutrient here, or the chemoattractant, or the oxygen. And chemotaxis is also can be viewed as a transportation of bacteria toward the nutrient. Another example here is the DDC bacteria, which we can found here in a wet medium. In the absence of nutrient, a specific characterization of this bacteria is the production by themselves of chemical to be aggregated. And this auto-production property can be found in the tumor growth and play a major role in the tumor growth or in the treatment in certain diseases. Biomathematical modeling is based on the combination okay, of three phenomena: diffusion, transport, and reaction. And a typical model describing chemotaxis is a Keller-Segel model. It is the most popular model to the chemical control of cell movement. The Keller-Segel system is the model of population dynamics, describing the evolution of the cell density and the evolution of the concentration of the chemoattractant. The cell density equation here contains two terms. The first one modeling the diffusivity process, and here A is the mobility or the function of diffusivity of the cells. And next here, the second term is represent here the chemotaxis term. This term represent the response of cells to the gradient of a high concentration given by the gradient of C, and C is the concentration of chemoattractant. This operator, it is we can see it at transport operator. It means here chi is the chemotactic sensitivity of cells, and cells are attracted or are directed according to velocity, and the velocity is here is minus the gradient of C. It is a transport equation, and cells detect this signal, and it is directed to the high concentration of chemoattractant. Okay. Here we have the sign plus. It is the attraction. If you take here minus, it's a repulsion phenomenon. Next, for the concentration of the chemoattractant, we have a simple diffusion equation. We have only a diffusion term here, and to model the production of chemoattractant, it is modeled here in this term. The rate given here by alpha, modeling the production of chemoattractant to attract the other cells, and here beta is the rate of death of the chemoattractant. We can add in this model two tensors, lambda and m, to specify is the medium where the phenomenon occur is isotropic or non-isotropic or heterogeneous. Okay. We are interested mainly in the numerical approximation or numerical resolution of this kind of equations. Just in few words, in mathematics in general, we are not able to calculate the exact or the analytic solution of a system of non-linear or linear system. Okay, so it is only for academic equation we can compute the solution. It's okay, but in general, for the real problem, it's not possible to exhibit exact solution. For that, we construct numerical methods to approximate the solution on meshes as fine as we want, and next we calculate the approximation of the solution on this mesh. For example, if you take this mesh with for example 500 triangles, it means that we are able to give an approximation of the solution at 5,000 point in this domain. and which give a very, very good approximation of the solution. Okay, we can compute at the center of the triangle or the center of the vertex of any polygons, etc. It's a big, very large domain in applied mathematics, how to construct a numerical scheme and how to solve this equation. Okay, to show the behavior here of the solution of Keller-Segel, we simulate here the evolution of the cell density, starting from the initial density, which equal 1 in the red square here in the center, and the chemoattractant are located here around the cell density in the 4 spot here. Okay, we start from the initial density. Firstly, we have the density of cells evolve by diffusion. Since the if you look here, the displacement, it is symmetric and uniform in all directions. It is a random distribution. It means here the diffusion contribution is important in the first stage in the evolution of the system. And next, after a short time, the cells detect the chemoattractant here, which are placed around the density center in this one. And here we see the influence of the chemotaxis operator and the transport of cells to the site here of chemoattractant. And the attraction continue, okay, when the time increases. Okay, it is the main phenomena for the Keller-Segel system. Okay. Now, the first example where chemotaxis play a major role is bone regeneration. We begin by, before writing the equations or the model in mathematics, firstly, we have to understand what is the biological process, and we begin by describing the biological process. This work is in collaboration with Yves Caudière and Zero. A bone is a living tissue which has the ability to regenerate by itself for small fracture. After a fracture, the 3 steps of bone healing process are, firstly, we have a hematoma step during 10 days. It's the first stage. And the second step is the bone regeneration. It is a ossification of the membrane. It is during 3 weeks. And the last step is remodeling of the bone. During a fracture, the rupture of blood vessels and bone matrix will cause here the release of chemical signals called here growth factor, and that will attract neighboring cell to the injury area. In this scheme, I show you the mechanism of the bone healing. The bone healing begins by, firstly, by the migration of mesenchymal cells. They are the stem cells. And these stem cells are present in the periosteum and in the bone marrow. And these stem cells migrate to the site of the injury. And next, stem cells here, in the presence of this factor, growth factor, differentiate into osteoblast. And next, this osteoblast mineralize the bone matrix to construct the bone. And all this phenomena can be occur or can be happen when in the presence of osteogenic growth factor, and it is a chemoattractant. It is a chemoattractant in our analysis. Okay, now we want to translate this this biological process in mathematical equations to try to solve the bone healing. Here we consider the model proposed by Bailan Plaza and Vader Newland, which take here into account the several mechanisms of the bone regeneration process. For the stem cells, we find here the Keller-Segel operator. Firstly, we have the diffusion operator of the stem cells, and the second one is the haptotaxis term, and the haptotaxis term here is due to the presence of the high concentration in the bone matrix, is 2 gradient of M. So here the stem cells are directed towards the region where the variation of the bone matrix is high. M is here the bone matrix, and which exactly the injury zone, or for example, where we have a fracture. Okay. And we have here the reaction term. We have firstly the mitosis, and we find here the differentiation of stem cells to be transformed in this term is transformed in osteoblast here, and rho here is a quotient of conversion from stem cells to osteoblast. Okay, this term here is negative. That means stem cells are here transformed on osteoblast. This is the differentiation process. And in this mechanism, because it is biological, we need to have the growth factor here, G, to ensure that the reaction happens. The osteoblasts here are fixed on the bone. Here there is no evolution term. It is a simple ODE equation. And the bone matrix here, we have the transformation of the osteoblast in bone with the term lambda B, and we have also the degradation of the bone by the term minus B. The osteoblast degrade also the bone matrix. Okay, and finally we have here the osteogenic growth factor, which is produced by osteoblast. Here we have the production of growth factor by the presence of the osteoblast. It is the auto production, okay, of growth factor due at the term to increase the healing of the bone. Okay, I give you only here a numerical simulation. We simulate here numerically the healing of skull fracture in rates. This simulation corresponds to an experience presented in the paper of Cooper and all. And here the defect is a whole fracture with a 2.3 millimeters. Okay, and we consider here the whole fracture is symmetric. For that, we simulate only the quarter of the domain. Okay, we take the quarter. This part is a quarter here, and the dark region here is a bone, and the white region is a fracture. Okay, the domain here contain only the bone, and we place here the two clusters along the broken way, the broken bone. The broken bone here is the friend between the dark and white region, the fractal and the hole. And initially, we have only stem cells and the growth factor. Okay, and we are placed near the fractal here, only between the dark region and the curved line, this one. The simulation of the skull fracture needs the construction of a complex mesh, and the mesh is refined here around the injury region, because all biological phenomena are happens here in this region, and we want we are able here to consider an accurate. We need an accurate solution near this region. Okay, for that we consider refined mesh about this one. Okay, we see here the bone healing after 3 days. After 3 days, we observe firstly the formation of osteoblasts, because osteoblasts are not initially present. We have the creation of the osteoblast, and these osteoblasts are created where the stem cells are initially concentrated. This osteoblast next synthesize in a new bone matrix, and we see here the formation of a new bone matrix. And we see here that the stem cells are moved towards the center of the fractal. Okay, after 42 days, here the stem cells disappear, and it is here in this figure. And in this case, we can consider that the healing is achieved. Okay, and we have here the final formation of the bone. It's given in this figure. Okay, and here we calculate the defect healing is approximately 42%. And here we say that there is no union fracture because the initial defect is too large. Okay, and we find that our results are very close to the article of Cooper to validate the model and the numerical approximation. Okay, now in the next part, we focus in breast cancer initiation. This work is in collaboration with François Fouché and Mustafa Ibrahim. Breast cancer is the second most type of the cancer after skin cancer. Okay, we are interested here in the initiation of the breast cancer by modeling the stepwise mutation from a breast cell, normal breast cell or healthy breast cell, to a tumor cell. In breast biology, the cancer stem cells hypothesis makes evident that mutation of an oncogene or tumor suppressor gene, TSG, are necessary to model the transformation of normal breast cell to tumor cell. And breast cancer here is driven by two classes of oncogene or of tumor suppressor gene. We denote here by TSG1 and TSG2, and which correspond to parents. Okay, the two TSG here are labeled TSG1 and TSG2, and next we denote by TSG1 plus plus and TSG2 plus plus. The oncogene where the two alleles are not mutated. Okay, in other term, we can write here, as I show you. We describe here the mutation, the step mutation pathway to explain the initiation of the breast cancer. We consider here the mutation diagram given in the paper of Enderling, and we consider this diagram of mutation. We denote firstly, we consider row I here the probability of mutation of muting one allele of every step of the mutation in this mutation diagram here, and we consider this this implication of the mutation. If you consider here row 1 is the probability of mutating one allele of TSG1. Okay, row 1, and next after this step, now we pass the second step. Is row 2, and row 2 is the probability to SG1 of muting of the second allele. Okay, and we consider now TSG1 is completely muted. And next we pass now to SG2, and to SG2 here we have the probability for SG2 TSG2 of muting of one allele. We have plus minus. And finally, row 4 is the probability, okay, for the two SCG are now completely muted, and we consider this cell is now consequence. To model in to translate this okay, this diagram in equations, we consider firstly 4 ODEs or ordinary differential equation here describing the stepwise mutation from the healthy breast cell to a tumor cell. When tumor cell are here, we consider now one PDE, partial differential equation. We consider here the evolution of the tumor by considering a Keller-Segel type system. We have one PDE equation to model the solid tumor growth, and it is invasion in the breast. And we have also one partial differential equation modeling here the matrix degradative of enzymes. We denote here NDE. And this NDE degrade the tissue upon contact to make space for the solid tumor to develop and grow into. Okay. We denote here F the fraction of the healthy breast cell, and we denote by U is the tumor cell, and produced by chemical here the enzymes V, which degrade the stem cells. Okay, to explain the mechanism. And now we write the equation for that. you write the equilibrium equation for each cell. Okay. We translate now. Okay. In terms of reaction equation or balance equation, the gene is mutation for this system. Firstly, we denote here. I give you the notation. The inactivation of a gene acquires two alterations. For that, we denote by Q here a cell alteration, or we talk about LOH in TSG1. Okay, and we note here by Q. Next, we denote by R as a cell TSG1 completely inactivate. And next, we denote by S the cell alteration LOH in the TSG2. Okay, for the first equation, it is the fraction of healthy breast stem cells. We write here a balance equation. We write the evolution of this fraction. We have here, firstly, you have the degradation. The degradation of healthy cell here is due of V, and V represent the concentration of enzymes. Okay, then healthy cells degraded in the presence of the enzymes. And here we see here exactly the alteration of this healthy cell by the probability ρ1 to be the first class of cells with alteration in TSG1. This quantity is exactly the same as this one. And now we have a diagram, and here a diagram here to pass from cells to another, from mutation to another. And now TSG1 here, when the second allele is mutated, you have minus ρ2 with probability ρ2, and we find this term in exactly in the term in R. And we continue for this class, the TSG1 minus minus, and next we obtain ρ2 and ρ4. And finally, after this transformation, when the mutation is complete, now we add here the tumor cell density, and we consider the cell is cancerous, and we add this contribution in the tumor cell evolution. Okay, for the tumor cell density, the evolution we consider here is the same as the same phenomena as Keller-Segel model. We have here the diffusion of the tumor cell, and we have here the apoptosis term, and this term is directed by the high gradient of the healthy cell. Okay, the tumor gradient is directed to the high gradient of healthy cell. Okay, and we add here the proliferation terms. For the enzyme concentration, we have simply a diffusion term, and we add here the production of enzymes in the presence of density, because here we have a plus alpha U, and U here is the density of the tumor cells. and we have also is a decay indicator. Okay, it is a complete model. I present to you, I show you only numerical result. In our numerical scheme simulation, we consider here that the breast is isolate, and so for that we consider there is no exchange between the breast and the outside of the domain. Okay, and we write here in mathematics, we say Neumann boundary condition. We use, as before here, the mammography of the breast to construct a mesh according to this mammography. And in our simulation, here we have taken, well, we do simulation with almost 18,000 triangles, which means the solution is very, very accurate. For the initial condition now, we consider to initiate the evolution of the concept. We consider only the initial condition with cells with the LOH in only the first oncogene, TSG1. And we consider is equal 0.3 only in this red square region. And we complete the initial condition of the stem for the stem cellular cells by taking the complement of this one is 0.7 and 1. Okay, and here there is the parameter. Okay, you have many parameters to do the simulation. We draw here the evolution of stem cells at here the time increasing, the time increase in this direction, and we see here the destruction. In the first row here we have the healthy stem cells, and we see the destruction of the healthy tissue in favor of the evolution of the tumor cell. Okay, we have a concordance between the evolution of the healthy stem cells and the tumor cells. Because tumor cells are attracted by the healthy cells, Okay, and here we have considered the breast is isotropic. It is homogeneous. That means the direction, the diffusion direction is the same in all direction, and it's a constant. To show, to illustrate the ability of our scheme to take into account the anisotropic of a medium, here we have considered anisotropic tensor in the diffusion, the anisotroper of diffusion and for the chemo attractant part. And in this simulation, we see here the influence of the breast nipple, okay, by taking an anisotropic in this direction. Okay, and we see the here I present here in the second row here the evolution of the enzymes concentration in time. Okay, that is all for this part. I have time. Okay. The last example here concerns the brain cancer, the glioblastoma. This work in collaboration with Flavien Alonso and Orient Serondour. Glioma multiforme, GBM, is the most frequent brain cancer. And only 5% of patients survive 5 years post-prognosis. In this work here, we are interested in the modeling and the simulation of the evolution of tumor brain. And we add here, we are interested also in the therapeutic response to the treatment. We add here the treatment by surgery, chemotherapy, and radiotherapy. And finally, we are interested how to calibrate biological parameters by using data. For example, the MRI here. Okay. As other examples, I begin here by explain in my words that the biological process for this tumor. The tumor growth of GBM, the glioblastoma, is based mainly in angiogenesis. And here the tumor cells promote angiogenesis because it enables tumor growth in hypoxic condition. I explain here in this scheme the mechanism of the tumor angiogenesis. Firstly, the endothelial cells carry out blood vessels, providing nutriment and oxygen in the brain. Okay. Due to the tumor growth, hypoxic tumor cells are induced by the lack of oxygen. And hypoxic cell produce, here's the production of pro-angiogenic factors, and these factors are the vascular endothelial growth factors, we denote VEGF, and this enhances the formation of new blood vessels. Okay, it is a mechanism here to explain the development of the glioblastoma. For the modeling for this problem, in the modeling we retain here 4 quantities: the density in tumor cells and the concentration in oxygen. Okay, it is in general in many papers it use this simplified model, but here in order to exhibit angiogenesis, 2 quantities are added into our model: the density of the endothelial cells and the concentration in the vascular endothelial growth factor. We are in the presence of 2 systems of Keller-Segel, and the 2 systems are coupled. Okay. It is classical here for the tumor cell density. We have the diffusion and the apotaxis cell due the high gradient of the concentration of the oxygen. And in this model, okay, this is a classical reaction term. We add here the treatment of this of the tumor. Okay, we have the concentration. We add here the consumption of oxygen by the tumor, and also for the density of the endothelial cells and VG. We have also a Keller-Segel system. We have the diffusion term, and the endothelial cells here are attracted to the high concentration in the growth factor of the vascular endothelium. Okay, and here it is equal. Okay, and we see here the linking between this model and the evolution of the tumor cell. Okay, for our numerical result, we use here MRI for patient, and we use here free databases. We have here, if you are interested by this database, and this databases we have the MRI sequence for many patients. And here we consider a patient with he's 60 years old and with a glioblastoma of 4.3 centimeter here. Okay, and it is the same. It's the same MRI, but in different sequence, because we can inject here gadolinium to show the tumor in different way. Okay. For our numerical solution, we consider here the original MRI, and next we construct a segmentation of the brain on the mesh according to this MRI. And in the domain here, the brain is anisotropic. The gray matter here is in dark blue, and the white matter here in light blue. And next, to impose the initial condition and take into account of this tumor, here we have three regions. The core here in green, it is the tumor. The edema here in yellow, and finally in red, it can be considered as we see that the flare of the tumor. Okay, I focus now to the modeling of the treatment. We take into account here surgery, chemotherapy, and radiotherapy are the treatment commonly used for GBM. Surgery is very simple in mathematical point of view. The tumor core here is removed, and we consider that all quantity, the density of the tumor cells, the concentration of the oxygen for endothelial cells, etc. All this quantity are removed, and we consider zero in the surgical area. In the other area, in the rest of the brain, we consider the same solution or initial condition. For chemotherapy, we use the protocol used in France, and here the drug commonly injected here is the temolizumab. It is TMZ. And in mathematical point of view, we add only the dose the dose administered by the drug, and if the chemotherapy is effective at time t, with this term multiplied by the dose of administration. And this term is a source term in the equation. And finally, for the radiotherapy, in France, it is recommended to perform radiotherapy 5 days a week for 6 weeks with some fraction. With here we consider 30 fraction of 2 grams per day. And to model this, radiotherapy works by sending a dose of radiation only at local position. So in this equation, we impose the radiotherapy only in the irradiate term. Okay. And for the model of radiotherapy, the model is very, very complicated because the efficiency of radiotherapy depends on a lot of parameters, depend on the dose administered, the duration of radiation, and this function over here we have many parameters: alpha, beta, mu, etc. Okay. Here I give you only the number of the triangles for this simulation. It is about 2,000 triangles. Here some data. The brain is not symmetric. The diffusion coefficients are not the same. We consider in the white matter here. Is that the coefficient of this diffusion is said to be 5 times faster than the gray matter. And in the region where in the post-surgical area, here there is nothing, and we set here the diffusion in DPS here to be 50 times faster than the gray matter diffusion coefficient. We apply here the protocol as we used here in France. We consider surgery is performed for our simulation at the first day of simulation. Okay, we start with that. And radio and chemotherapy start both in 2 weeks, in 14 days after the surgery, and last for a period of 6 weeks. And chemotherapy is administered every day. And okay, radiotherapy administered periodically is okay. And in general also the radiotherapy is administered locally, and also around the area region, 3 centimeter area bordering the whole tumor area. Okay, I show you here the number, the initial condition, the number of tumor cells, the number of endothelial cells. Okay, it's not the same in gray and white matter. Okay, for the concentration for oxygen and VAG. Here I present you only this graph for this numerical simulation. In the y-axis here we have the number of tumor cells in the brain. This is a global number, the global number of the total number of cells versus time. And here we have do many simulation with the first one. We do nothing, no treatment, and here we do surgery TMZ is chemotherapy, radiotherapy, and the combination with the chemotherapy, radiotherapy, and also we add surgery. The first comment is the curve is here. No treatment and chemotherapy are exactly or very, very close. And so it's difficult to distinguish at this scale. Is this curve in orange? Okay. No treatment and TMZ are the same. In this conclusion says that for all the patients, it's not necessary to give chemotherapy only because you have the same result as do nothing. Okay. Next, you take surgery. Surgery is good, okay, because the number of tumor cells decrease, and after that continue to increase. Okay. If you take radiotherapy is very efficient. Okay, in this period, here we have radiotherapy. Okay, and but when we stop radiotherapy, the number of tumor cells increase. And if you combine chemotherapy and radiotherapy here, the result is better. But when we stop the treatment, we have also the number of tumor cells increase. The best here treatment is to combine surgery, chemotherapy, and radiotherapy. We see here in this point that when we combine, this curve is better than the other. Okay, the number of tumor decrease, and when we stop here after I think 56 days, the treatment, the number of tumor cells remains constant. Okay, there is no decrease, no increasing as before, and for this tumor, the combination of the three treatment is good, and this protocol it seems good. Okay, I have perhaps I take 2 minutes, 3 minutes to explain.
Okay. Yes.
Yeah. Okay. Thank you. To do our numerical simulation, we have used parameters. Okay, and we have very many parameters in the literature. We take, for example, this value by considering here this article. Okay, it's very and you can see that this parameter depend on the referee. And the question which we ask here, are these parameters are true, and what are the right parameters? If you take you have a patient, what are the parameters? Okay. Okay. We don't know how to find the true parameters for a patient. For example, if you take here the diffusion coefficient for the tumor, the value is very different. It depends on the water. It is the same units, but it is, if you take the 10 power minus 11 and 10 minus 8, the spread of tumor is not the same. It's not the same. Okay. And so we are interested by the question, what are the right parameters? Here in this project, we try to calibrate the state variable, meaning the unknown of a model, and also the parameter of the same model. Okay. The same parameter is not, we don't know exactly what it is. Okay, we have a construct here, a model, and the model for the numerical simulation produce the same phenomena. Okay, we say that the figures here are the same or very close. Okay, we have a dot here on our model. Our initial conditions are not good also, and we don't know the parameters. And here we want to use real data to ameliorate or to calibrate our model, the parameters and the initial condition. And this problem is complicated. That's if you want to use real data, for example, MRI, you have some MRI at, for example, T equals 0, at T after 6 months, 15 months, etc. And we need here to build a method that works with sparse data over time. Okay, because we have we don't have the information at any time and also at any space, because if you have MRI or analysis, it's not possible to have the value of the locally the value of the cell density, of the concentration on oxygen, etc., and the parameter. Okay, we have many errors here. It is this problem we have to solve an inverse problem to calibrate the parameter and the model. Here we try our method by using a simplified model of GBM. We consider only the concentration of the oxygen in the tumor cell. And in mathematical point of view, we aim to find the parameter only for the reaction term, rho, delta, alpha, beta, gamma. And here in this method, we suppose that the diffusion coefficient and the chemotactic coefficient is given. Okay, we start by to validate our method. We consider, we look for this parameter theta exact here. For example, we give this number 0.2 for rho, etc. And we compute the numerical solution associated to this parameter. Okay, and here the exercise. Now we forget the theta exact, the parameter, because in general we don't know what is the parameter. But we know here the observation. For example, it's equivalent to MRI. We have the solution, U and C at any time and in each point in the space. And in this part, we don't know what is the parameter. We have only the consequence of this simulation. It's exactly the definition of the inverse problem. You have here the solution, but we don't know what are the parameters. Okay, and we consider here the observation, or if you want, are the solution computed by this exact solution. Okay, and we start here by initial guess theta 0. It's not theta exact. It's different from this one, and we solve our inverse problem to construct a numerical sequence for all parameters. And the parameter here, we say that by solving this problem, we find the parameter, the expected parameter here in the continuous line. Okay, but this problem is an open problem for us, and to apply this inverse problem or the calibration for the glioblastoma, it's not easy to do that because we have many missing data. Okay, here it's possible to do that because we have the exact solution. Okay. In conclusion of this talk, our method or our numerical method and modeling here relies on solving partial differential equations system which corresponding to the evolution and the treatment of the certain cancer. This system reproduces the behavior of the biological process, and but the behavior of the solution depends on several parameters. In personalized medicine, we aim to adjust the biological parameter using the available data. And in our approach, we use only, here's a numerical solution of a deterministic system. Okay, and the right parameter can help predict the development of the cancer and therefore adapt the treatment according to this prediction. Okay, it is our aim, but okay, we try to give a short answer of this problem. And thank you very much.
Thank you very much for this interesting talk. Well, it's time for questions. I think we can receive a few questions. So any questions from the audience?
Okay, they can write by chat the question.
Either by chat or raise hand to ask the question directly, or unmute themselves and ask the question directly.
Okay.
Well, I don't see any raised hands.
Okay.
Well, okay. So, okay, I have one raised hand. Professor Talhou, can you please unmute yourself?
Hello, everybody.
Hello.
And thank you for this interesting project, for all what you want to present in this series of seminars concerning mathematical biology. Thank you, Professor Mazen, for this nice talks and very rich. I have just one question concerning the mathematical systems modeling the breast cancers.
Yeah.
I see that it is very complicated and is performed by coupling many differential equation, ordinary differential equation with PDEs.
Yeah.
With Keller-Segel model, I guess.
Mm-hmm.
And you have talked about numerical results. My question is concerning what about mathematical results, and particularly maybe a global existence. Do you have this?
Okay, in the work with François Souchet and Mustafa Ibrahim, in fact, we have constructed a finite volume scheme, and we show that this scheme is convergent. And for that, we obtain the existence of the global solution of this system.
Okay, and what about the regularity and
The regularity is very low. Here we are considered the degenerate problem, and that means the chemotaxis function chi vanish at 0 and 1, and so we have the density is between 0 and 1, and but the concentration of the concentration of enzymes is positive, and the solution is in L2H1, and it's all.
Okay, and this means that you have the maximum principle.
I have the maximum principle for all quantities. I can write it.
And what numerically also you can verify the maximum. We can preserve the maximal.
I preserve. We choose our scheme preserves the maximum principle, and we show here. I think I put here, and here we have, for example, V is between 0 and 1. F. We preserve the maximum principle for all quantities, and U here is between 0 and 1.
Good. Okay, in the same mood, what about the system appearing in glioblastoma? Because here I see that you have indicatrix function in the system, and maybe you have some singularities. Do you preserve also mathematical results? Do you get mathematical results on the system?
No, it is only in
glioblastoma.
It is only in the source term. It is only here, and this term or in L infinity. Okay. And the regularity of the solution is only A by gradient of U in L2H1. Okay.
Okay.
It's not possible. If you want to more regularity, you have to derive this equation, so it's not possible.
Okay,
but the minimum regularity is enough for us to do numerical solution and to show the existence.
Okay, this means that you have used semigroup theory result for second number in L infinity.
Yeah.
Okay, thank you.
Thank you for you.
I see a question from Professor Wool. Thank you for this very interesting talk regarding modeling of the brain tumor. Are there any assessments for the coupling of mechanical stress and diffusion chemotaxis on tumor development? So, do you have any idea whether there are any assessments, any evaluation, or possible coupling between mechanical stress and diffusion chemotaxis in this case?
Yeah, I think for brain tumor, the mechanical stress is not it's not important, but the mechanical stress is very, very important when you treat the bone regeneration model, because the fracture in general you have to fix it to have the regeneration of the bone. But I think in brain tumor, no, there is no constraint here, because the tumor is only in the matter, in the white matter. Okay, there is no constraint, real constraint here, I think.
Well, probably there is, I don't know, probably there is a possibility for some kind of coupling of mechanical stress, probably because of the fluid, if you have an increase in the volume. I don't know. I think this is an interesting question, but
Coupling, in fact, it's always possible mathematically, but the problem is the study after, if it's still
it's useful or not.
possible and easy or not, because coupling with elasticity or stress equation maybe make appear more mathematical problem and relevant problem.
Well, we have a question from Professor Nassif. Can you please unmute yourself?
Yes, I am unmuted though. Can you hear me?
Yes, we can hear you.
I hear you.
Okay, good. Thank you, Professor Saad. It's very interesting lecture. I couldn't really see all your slides because I had problems with sharing with reading your slides. But I read the first one, and so you have essentially a system of with two variables, which are U and C, right? This is
Yes,
and the involvement of these functions is through some sort of elliptic operator, and yet it is you have like two parabolic equations.
Yeah, good.
Which are not non-degenerate. That's right.
No, it's a degenerate.
They degenerate. I mean, the coefficient can go to 0 or.
No, the coefficient here vanishes at 0 and 1.
Ah, okay. So this what deteriorates the regularity then.
Yeah, so for the regularity, and it is also when the chemoattractant and vanish at 1, it means that the transport is vanished when the density of the tumor is maximal. We consider in the area here, it is we say here is a volume filling effect. We consider if the volume is comment on dit complet?
Speak in French.
Yeah. Ah, alors, ben, en fait, c'est le volume, le filling volume effect, c'est-à-dire on dit que les cellules vont se déplacer, mais par contre, si on suppose qu'on a une densité maximale pour ces cellules, ben après, on ne peut plus se déplacer vers cette zone. Et donc, ça annule en fait les termes de diffusion, le coefficient de la fonction de diffusion et aussi la fonction de sensibilité. Donc, c'est pour cela que ben, la tumeur arrive, elle est attractée, elle est maximale, elle ne bougera plus. Après, elle va se déplacer uniquement par diffusion.
Donc, il y a des phénomènes de singularité donc, sur les bords.
Après, si on prend par exemple, la fonction U qui dépend que de U, on peut considérer que les solutions, souvent les solutions, elles explosent en temps fini. C'est-à-dire parce que ce terme, en fait, il continue à attirer les cellules et donc U, ça va tendre vers l'infini. Donc, on perd le principe de maximum.
Oui, mais à ce moment-là, comment on fait numériquement s'il y a des phénomènes d'explosion? Comment on contrôle?
Comment on fait? Comme on peut. On régularise en général autour et numériquement, on est obligé de tronquer à un certain moment la solution.
D'accord.
D'accord. On dit que les cellules, quand même, numériquement, il va être à un maximal et on considère à partir de cela, c'est-à-dire la solution, elle a explosé.
Très bien. Merci. Thank you.
Thank you. Any other questions from the audience?
In Arabic now.
Well, just take a general question. I don't want to go into mathematics at the end. Do you have, well, we saw that you're exposing many mathematical models. What is the use of, is there feedback from medical doctors about your results? Is there usually a communication whenever you're doing such a work?
Okay. For the last one in glioblastoma, because This work is in collaboration with a biologist, which work here in the hospital, and we discuss with doctor to validate this problem.
So there is always kind of continuous communication between the modeling people and the medical or biologists.
Yeah, our aim is to use MRI to adjust the parameter. If you have the good parameter for the patient, because it's different, and if you have a good numerical simulation, we can after perhaps give the best treatment for the patient. If you have the good parameter, we say we can change the protocol, for example, and we see what is if the tumor grow or not, etc. This is our aim in general, but it's complicated.
Yeah, it's not that simple. This is a long-term goal, probably.
Yeah.
Okay, I think we can stop here if there are no more questions. Thank you everyone for joining. Thank you, Professor Saad, for this very interesting and informing lecture.
Thank you.
And looking forward to more and to have you, to meet you everyone in future talks.
Okay,
Thank you everyone.
Thank you. Bye.
Bye.
Bye.